unlocking genetic innovation, U.S. DNA synthesis market to hit $4.62 billion by 2033. The U.S. DNA synthesis market is on the cusp of a revolution, projected to skyrocket from a $1.25 billion valuation in 2024 to a staggering $4.62 billion by 2033, growing at an impressive clip of 15.93% per year. This meteoric rise isn't just about numbers. It's about the transformative power of DNA synthesis in synthetic biology, personalized medicine, and biotechnology research. But here's where it gets controversial. As gene editing technologies like CRISPR-Cas9 become more widespread, the demand for custom DNA sequences is skyrocketing. This need is fueling the U.S. DNA synthesis market, but it's also raising ethical questions about the limits of genetic manipulation. And this is the part most people miss. While the technology is advancing at lightning speed, regulations are struggling to keep up, creating a gray area that could lead to misuse. But let's not get ahead of ourselves. Imagine a world where DNA synthesis is so precise and affordable that personalized medicine becomes the norm. This isn't just a sci-fi dream, it's the future we're heading towards. But are we ready for the ethical and regulatory challenges that come with it? Share your thoughts below. And for those who want to dive deeper, here's a snapshot of the U.S. DNA synthesis market. The U.S. DNA synthesis market size was estimated at $125 billion in 2024 and is projected to reach $104.62 billion by 2033, expanding at a CAGR of 15.93% from 2025 to 2033. This high growth is driven by increasing demand in synthetic biology, personalized medicine, and biotechnology research. Advancements in gene editing technologies and declining costs of U.S. DNA synthesis are further accelerating market expansion. The U.S. remains a key player in global genomic innovation, supported by strong investment in R&D and a well-established biotech infrastructure. Advances in gene editing technologies, particularly CRISPR-Cas9, are revolutionizing the field. These tools rely heavily on synthetic DNA sequences such as guide RNAs and donor templates to target and modify genetic material precisely. U.S. DNA synthesis enables researchers to design and manufacture these sequences quickly and accurately, which is critical for the success of gene editing experiments. As the demand for high-throughput genome editing increases in academic and commercial research, the need for custom DNA sequences grows in parallel, directly fueling the U.S. DNA synthesis industry. The applications of CRISPR and other gene editing technologies are rapidly expanding across different fields. The demand for precisely synthesized DNA sequences increases as these technologies gain wider adoption in research and commercial sectors. This need is driven by the requirement for high-quality, custom DNA to ensure accuracy and efficiency in gene editing experiments. As a result, the growing use of gene editing tools directly boosts the U.S. DNA synthesis market. The rising demand for personalized medicine and gene therapies propels the U.S. DNA synthesis market. Customized treatments are tailored to individual genetic profiles, requiring the quick and accurate synthesis of DNA sequences for diagnostics and therapies. This method is vital in developing gene treatments for rare genetic disorders and cancers. Being able to synthesize specific DNA sequences allows for creating personalized therapies that are more effective and cause fewer side effects than traditional options. Recent advancements highlight this trend. For example, in May 2025, Doctors at the Children's Hospital of Philadelphia and the University of Pennsylvania successfully treated an infant with a severe genetic disorder using customized gene editing therapy. This groundbreaking procedure involved base editing to fix a genetic mutation, marking an important milestone in gene therapy. Such developments emphasize the vital role of U.S. DNA synthesis in enabling personalized treatments tailored to the unique genetic makeup of each patient. The U.S. DNA synthesis market is marked by a dynamic convergence of well-established expertise and innovative newcomers, intensified by active mergers and acquisitions, strategic alliances, and breakthrough product launches. As precision medicine, gene editing, and synthetic biology applications expand, companies that combine technological excellence with scalable, ethical, and cost-effective solutions are poised to lead the market's evolution. Emphasis on quality, speed, data security, and regulatory compliance will remain critical in sustaining long-term growth and maintaining customer trust in this rapidly advancing industry. Key players in the U.S. DNA synthesis market include Thermo Fisher Scientific, Twist Bioscience, Bioneer Corporation, Eaton Bioscience, LGC Biosearch Technologies, IBA Life Sciences GmbH, Eurofins Scientific, Integrated DNA Technologies, Quintera Biosciences, and GenScript. This report forecasts revenue growth and provides an analysis on the latest trends in each of the subsegments from 2021 to 2033. For the purpose of this report, the market is segmented based on service type, application, and end use. Service type: oligonucleotide synthesis and gene synthesis. Application: research and development, diagnostics, and therapeutics. End use: biopharmaceutical and diagnostics companies, academic and research institutes, and contract research organizations. The oligonucleotide synthesis segment led the U.S. DNA synthesis industry in 2024, driven by its vital role in research diagnostics. The gene synthesis segment is expected to grow at the fastest CAGR of 17.62% during the forecast period. The research and development sector led the U.S. DNA synthesis market in 2024, capturing a significant revenue share of 63.82%. The therapeutic segment is expected to have the fastest compound annual growth rate, CAGR, of 17.13% during the forecast period. The biopharmaceutical and diagnostics companies segment led the U.S. DNA synthesis industry in 2024. As the U.S. DNA synthesis market continues to evolve, companies that can navigate the complex landscape of technological advancements, ethical considerations, and regulatory compliance are poised to lead the way in this transformative industry. The future of DNA synthesis holds immense promise, but it also requires careful consideration of its implications on society and ethics. Share your thoughts on the future of DNA synthesis and its impact on personalized medicine, gene editing, and synthetic biology below. Exploring the booming gene synthesis market. Trends, key players. The gene synthesis industry is exploding, but are we ready for the ethical storm brewing beneath its rapid growth? With a projected 16.1% annual growth rate through 2032, this market isn't just reshaping biotechnology, it's rewriting the rules of life itself. Let's unpack why this sector is skyrocketing, who's leading the charge, and what controversial questions we should be asking about our DNA tinkering future. From lab curiosity to life-saving revolution, gene synthesis, the artificial creation of custom DNA sequences, has evolved from niche science to the backbone of modern medicine and agriculture. Imagine researchers designing cancer-targeting genes like architects blueprinting skyscrapers, or engineers creating drought-resistant crops, nucleotide by nucleotide. This isn't science fiction. It's why industry giants like GenScript, Thermo Fisher's GeneArt, and IDT are racing to dominate a market expected to hit billion-dollar milestones within this decade. The growth engine, more than just petri dishes. 3 seismic shifts are fueling this boom. 1. The personalized medicine earthquake. Tailored cancer therapies using a patient's unique genetic code, think of it as bespoke DNA tailoring, are pushing demand through lab ceilings. 2. The CRISPR domino effect. Gene editing tools have created a voracious appetite for synthetic DNA, the raw material for everything from COVID vaccine development to de-extinction projects. 3. Agricultural Armageddon aversion. With climate change threatening food security, synthetic genes for pest-resistant crops have become agriculture's new insurance policy. But here's the controversial twist. As companies like Eurofins Genomics and GeneWiz make gene synthesis cheaper and faster, are we sleepwalking into a bioprivacy crisis? Your synthetic DNA blueprint could become the ultimate corporate asset or security vulnerability. Market segmentation, the gene size gold rush. The industry splits its ambitions into 4 DNA length categories. Under 1,000 base pairs, the quick fix genes for routine lab work. 1,001 to 3,000 BP, the sweet spot for most therapeutic proteins. 3,001 to 5,000 BP, complex gene clusters for metabolic engineering. And 5,000 plus BP, frontier territory for synthetic chromosomes, the corporate genome war. The competitive landscape reads like a biotech Avengers roster. GenScript, the Swiss Army knife of gene services. GeneArt, Thermo Fisher, the luxury sports car of precision DNA. IDT, the people's champion for academic labs. DNA 2.0, the mad scientists pushing synthetic biology's limits. The elephant in the lab. While the report celebrates growth, it glosses over the industry's dirty secret. 30% of synthesis time is wasted fixing errors in artificial genes. This inefficiency gap has created a gold rush for AI solutions. Companies now use machine learning to predict DNA assembly errors before they happen, like spell check for genetics. Controversy alert! Who owns synthetic life? As startups like SBS Genetech and BioBasic democratize gene design, we're facing unprecedented IP battles. When a synthetic microbe cleans oil spills or produces insulin, who profits? The company that designed it? The patent office? Or humanity at large? This isn't hypothetical. It's already sparking lawsuits that could redefine biotechnology law. The innovation paradox. The report's rosy projections hide a brutal truth. While AI and blockchain promise revolutionary changes, most labs still rely on 20-year-old PCR techniques. The real growth driver? Old-fashioned human ingenuity, like the unnamed company that slashed production time by 30% through analog process tweaks, proving that sometimes the best solutions don't come from algorithms, but from lab-coded problem solvers. Your turn to weigh in. As we stand at this genetic crossroads, let's get controversial. Should there be open-source DNA databases to prevent corporate monopolies on synthetic life? Are current regulations sufficient when a high schooler can theoretically order pandemic-level pathogen genes? Does the promise of ending genetic diseases justify the risk of engineered biological weapons? Drop your thoughts below. The future of gene synthesis isn't just shaped by billion-dollar companies, but by the ethical choices we make today. Will this technology save humanity, or have we finally opened Pandora's petri dish? DNA and gene cloning market. Boom in gene therapy CRISPR innovation, 2024 to 2034. The DNA and gene cloning services market is booming, and it's easy to see why. With a projected value of $11.7 billion by 2034, this industry is revolutionizing how we approach medicine, agriculture, and scientific research. But here's where it gets controversial. As the demand for personalized medicine grows, so does the need for complex gene constructs and scalable gene synthesis. This has sparked a race among biotech companies to develop innovative cloning technologies, with key players like Thermo Fisher Scientific and GenScript leading the charge. But what exactly is gene cloning, and how does it work? In simple terms, it involves combining genetic material from two different sources to create a single DNA molecule. This recombinant DNA is then introduced into a host organism, which produces genetically modified offspring. This process is used in a variety of applications, from protein expression to gene function analysis and the development of gene-based therapies. The standard workflow for gene cloning involves isolating DNA from a source organism, identifying the target gene, and replicating it through molecular cloning techniques. Key procedural steps include plasmid DNA extraction, enzymatic digestion using restriction enzymes, and the insertion of recombinant DNA into a host cell to enable replication. PCR plays a crucial role in amplifying DNA sequences throughout this process. Gene cloning can be performed through two principal methodologies, in vivo and in vitro. Numerous human genes have been successfully cloned into microbial systems such as Escherichia coli and yeast, facilitating the large-scale in vitro production of human proteins. Currently, over 100 therapeutic drugs are manufactured using genetically modified cells, including E. coli, yeast, and mammalian cells engineered with human gene sequences. Gene cloning has emerged as a transformative tool in biotechnology, significantly contributing to advancements in human healthcare by enabling more effective treatments for chronic conditions and subsequently improving life expectancy. In the agricultural sector, gene cloning continues to enhance crop quality and nutritional content. The global market for DNA and gene cloning services is expected to witness sustained growth in the coming years. This upward trajectory is driven by the increasing adoption of gene therapies and ongoing innovations in DNA cloning technologies. Rising investment in research and development, along with growing demand for personalized medicine, further accelerates market expansion. Additionally, the sector benefits from robust funding support, continuous technological improvements, and an expanding number of companies entering the market. Despite these favorable developments, the gene synthesis and cloning market faces certain challenges, including a shortage of skilled professionals and protracted regulatory approval timelines. Nevertheless, ongoing technological advancements, strategic collaborations, and mergers and acquisitions in the biomedical and healthcare industries are poised to create substantial growth opportunities. With strong capital investment and increasing integration of gene cloning technologies into life sciences research, particularly in North America, the market is positioned for continued development. So, what do you think? Is the future of medicine and agriculture in the hands of gene cloning? Or are there potential risks and ethical concerns that need to be addressed? Let us know in the comments below.